A very good morning. I bid to all of our respective lecturers. Today I'm here to present about my research proposal on the title of Evaluation on the Effects of Ketum on Pain Tolerance among Ketum users in Malaysia by using the cold pressure task. Before I proceed to my presentation, let me introduce myself first. My name is Shani Radhakrishnan. My matrix number is 184202. Firstly, the introduction part. The Ketum leaves is an indigenous plant of the Southeast Asia, reported to be commonly used for its medicinal value and as a substitute for illicit opiates. Traditionally, these Ketum have been used for various medical conditions such as to treat muscle pain, intestinal infection, and other conditions. The statistic in Malaysia states that about 97.1% of Ketum users are Malay, whereas only 2.2% of Chinese and 2.8% of Indian and others. Next, why this research focus on pain tolerance is because, as we know, pain has been associated with many diseases. However, there are huge number of commercially synthetic drugs are available. However, this drug reported to possess adverse side effect to the consumers. And next, this research involved a specific method called pressure test, which useful to mimic the effect of chronic pain and also measure the pain tolerance among Ketum users. Next, the problem statement is the various studies have been reported that pain relief is one of the reasons for the Ketum use in the southeast region of Asia, such as Thailand and Malaysia. There are experimental animal model studies have suggested these possible analgesic properties of Ketum. However, there are lack of human laboratory study to date supporting such claim. Therefore, an experimental study using empirical methods to determine and evaluate analgesic properties of the Ketum is needed by measuring pain tolerance among Ketum users using a simple experimental procedures which is known as cold pressure, in order to establish that Ketum can be recognized as one of the traditional and alternative anti-nociceptive drugs. Next, I proceed to the objective. The general objective of this research is to determine the effects of Ketum on pain tolerance by using the cold pressure task. Next, specific objective is to evaluate the plasma concentration of mitragynine in participants before and after ingestion of the Ketum or placebo drink, and to differentiate the time taken for the participant to keep their hands immersed in cold water after ingestion of the Ketum or placebo drink. Next, the hypothesis is there is a significant difference on pain tolerance between participants who receive Ketum juice and participants who receive placebo juice. Next, these are some of the articles that I reviewed to do this research proposal. So the main point is that the main active alkaloid of the Ketum is the mitragynine. This is responsible to show the possible analgesic properties. And it also has been stated that at low dose, this particular Ketum can stimulate the brain and also can cause sedative-like effects at higher dose. Next, I proceed to the methodology. Firstly, the participant in this proposed study will be male who reported drinking Ketum juice more than once a day or daily for the past 12 months, and they must be at least 18 years of age when consent is obtained, and they also need to be complete 11 years of formal education. If the participant experience ongoing acute and chronic pain or neurological or psychiatric illness need to be excluded from this study. And this is a within subject design where the participant will serve as their own control, so there is no control group in this study. A simple random sampling technique is used to recruit 20 potential respondents for this study. Next, firstly, I will explain about how the drinks, either Ketum or placebo, will be ingested by the participant. Prior to 12 hours of the test, the participant not allowed to drink the Ketum juice. Before they receive the drinks, researcher will check for the sign of the withdrawals using the validated Ketum withdrawal scale. The participant will receive the first drink at first hour, for example 7 a.m. and another drink after 1 hour. If the first drink served as was Ketum drink, the second drink served will be placebo drink, and this will be vice versa. And the dosage will be used is 0.8 mg per kg, and there are 4 sequences of the drinks will be served. After ingestion, the cold pressure task will be performed immediately. So in the cold pressure task, the participant will first immerse their right hand in the 32 degrees Celsius water bath for 1 minute. After that, the hand temperature will be measured before they immerse the same hand up to the wrist into the 3-degree cold water bath. They will keep their hand in the water bath until the participant feel too uncomfortable or for 5 minutes maximum. And then the time taken in second, the participant take to immerse his hand until he removes from the water bath will be recorded. This okay. And next, the last one is about 1 ml of blood sample will be collected from the participant immediately after each trial of the cold pressure task in order to determine the level of active compound of ketum in the plasma. So in this research, the participant will be sorted by using randomization technique, and the participant and researchers will be blind to the drinks. The order of the drinks, either ketum or placebo, received by the participant will be randomized to one of the 4 sequences as shown in the table below. Next, the data collection in this research will be analyzed using repeated measures analysis of variance within group and univariate parametric analysis, which will express as a mean standard deviation of the emission time, where the p-value less than 0.05 is considered to be statistically significant. Lastly, expected results from this research is participants will be able to keep their hands immersed in the 3-degree cold water bath significantly longer, which is more than 3 minutes, particularly after ingesting the ketum drink compared to the placebo drink. If the ketum drink does indeed possess analgesic properties, or the mitragynine content in the prepared ketum drink is sufficient to produce any pain relief effect. These are the references that I have used to do this research proposal. That's all. Thank you.